All right. Hi, everyone online. Thank you for joining us and welcome to the One Health Academy. We've just done introductions in the room, and I'll go ahead and start off by saying thank you so much to the One Health Commission for sponsoring our webinar. For those of you enjoying the comfort of your own home right now and joining us, it wouldn't be without their help that we had this capability. So thank you to the One Health Commission, and also to ASM for those of us in the room. They graciously sponsor us every month, so we are grateful for both groups. And yes, thank you for all the support. I will go ahead and start introducing our speaker for tonight. Lori Miller is an environmental engineer with more than 20 years of experience designing and implementing systems to remediate and protect the environment. She has spent the past 15 years with USDA, where she currently serves as senior staff officer focused on planning, preparedness, and response activities related to animal disease outbreaks in the areas of waste management and decontamination. Please join me in welcoming Lori Miller.
Thank you so much, everybody in the room and on the phone. It's my pleasure to be here. So basically, I spend my days thinking about what to do with dead things. And I didn't grow up wanting to be a carcass disposer, but here I am. Here we all are. I work a lot in that space of, if there's an outbreak and the response is to, contain that outbreak, and often with African swine fever, which, is this double-walled hemorrhagic virus that has high mortality rates. If we were to get it here in the US, we would have massive animal mortalities. And the challenge with that is what to do with all of the infected, contaminated material then. And so certainly, ASF, it doesn't infect humans, it's an animal virus. And so that's certainly within the, animal health part of the One Health continuum. And the waste management part of it clearly affects the environmental leg of the stool. But indirectly, waste management, carcass management for ASF has a significant impact on human well-being and health as well, in the sense that, pork is a human food product, and the economic impact to producers and to the US economy is pretty significant. So it turns out that pork production represents 0.2% of our entire GDP. That seems like a lot to me. How do I get it to go forward? So I
just arrow down, yeah.
Okay.
Okay, need to go up.
Okay. So just to talk about, what this might look like if we were to have an African swine fever outbreak in the US. Taking North Carolina as one of the major swine production states, and these dots on the map here are from the National Agricultural Statistics Service inventory, where each dot represents 20,000 hogs or pigs. And so you can see in that region of mid-eastern North Carolina, there's a very high density of pork production. And so this is an image zooming in on one of the counties in that area, and just a random 16 square mile area has 18 production facilities in that vicinity. And I point this out to relate it to the strategy, the international strategy of responding to animal disease outbreaks, where if there is a positive premises, there's a 3-kilometer radius put around that for active containment and control, and then a 10-kilometer radius around, further expanding beyond that. And so imagine if we have a, so it's a 6.2 mile radius, how many production facilities there would be if there was a single infected premises in this region of North Carolina. And so what does that mean? It's not unusual in Duplin County of that state, where they have 2 million head on 600 farms. You divide that out, it's about 3,300 animals on average per farm. It's not unusual to have 2,500 sows at a single farm at 400 pounds each, 500 tons of material per premises to dispose of. And so the first thing people think of is, oh, well, we'll just bury it. And if you did that and laying the pigs end to end, you'd have a 2.6 mile long trench, and it would generate 40 gallons of leachate per day. And the leachate is about 2 to 4 times more concentrated than the lagoon slurry that is generated from these swine production operations. So it takes some careful thought before jumping to the conclusion that burial is the way to go. And in fact, if you look at the soils map for North Carolina in this vicinity, the Natural Resource Conservation Service has a soil survey where they map pretty much every county in the US by soil type. And they have a website, a model that you can determine, is the soil suitable for mass animal burial? And if it's not, it comes up red. If it's somewhat suitable, it would be yellow, and if it's suitable, then it would be green. And so you can see that this region of North Carolina, where they have all of the intensive production, is not suitable for burial. So it's very limited in that region. So let's talk about what are the options and the pros and cons, and how those would affect human and environmental health as well. So, there are a number of off-site options, such as incineration, landfill, and rendering, but that kind of flies in the face of the whole idea of quarantine, where you want to keep things contained. So then you look at the on-site options, such as open burning in pyres, deep burial, composting, and a sort of an innovative technology that we're working on now, which is above ground burial. And then there are also some ancillary activities like storage and transport that come into the equation. So if we think about incineration, so those offsite options are really good because they're designed for waste management. So they already have environmental protection built into them. The process gets extremely hot, 1,500 degrees Fahrenheit, and the process involves basically packaging the waste material into these Gaylord boxes, which are about a cubic meter, and then the exterior of the box is disinfected, loaded on a truck, taken to the waste to energy plant, and where it's picked up and tossed into the hearth with one of these giant grapple things, and it's burned at 1,500 degrees Fahrenheit, which is hot enough to kill pretty much everything. So that would be a great option. The problem is there aren't very many of them in the country. They're often used for municipal waste disposal near large municipalities, and large municipalities and pork production usually aren't co-located. So you might have to drive quite a ways to get to one of these plants. But having said that, we have done some work with waste to energy plants in both Indiana and New York. So these pictures were from the Indiana exercise, where they went into an egg laying facility, and they worked out a deal where they were able to exercise with the spent hens that had to be disposed of anyways, and they exercised this process. So it worked out really well, but as I said, there's limitations with it. So it could take some time depending on how far you are from the incineration plant. It could be relatively expensive because the transportation cost and having to package, I mean, can you imagine? So one of the largest hog farms in the US is 170,000 head farm in Texas. So can you imagine putting 170,000 pigs into little boxes and hauling them someplace? Yeah, so that would be expensive, time consuming, and but very low risk. I mean, once these boxes, you can get them double, triple wall, you know, with liners, and it can be very biosecure, but not necessarily cost effective. So landfill is another option, again, designed to accommodate waste materials. The landfill process is basically the, you know, waste is placed into these large cells, which are lined with plastic and or clay liners. The liquids that are generated as the waste decomposes are collected. That's called leachate. That's collected at the bottom of the landfill, and it's shown in this diagram here. The process of decomposition is anaerobic, so it generates methane and hydrogen sulfide gases, and the gases are controlled and often used as natural gas to for power or what have you. So the problem is that there is concern that if the landfill were to take infected carcasses, that the leachate might still contain the active virus. And the question is, how long will it take for that leachate to get to the bottom of the landfill, to the leachate collection system? And when it gets there, will the virus still be active? And that matters because the leachate is then either sprayed back onto the surface of the landfill, or it's discharged to oftentimes holding ponds on the farm, I mean, on the landfill site. And those holding ponds are similar to wetlands, and they attract wildfowl. So especially in the case of high path avian influenza, that was a huge concern. And similarly, it would be a concern for African swine fever, with feral swine possibly contacting those pond areas. So we have had some concerns by the landfills for that reason in terms of accepting the material. We're working on a risk assessment with our Center for Epidemiology and Animal Health at APHIS, which is nearly complete and is showing a low to negligible risk of reinfecting wildlife from the landfill leachate. But it's a complicated issue to communicate to folks who have the perception that it's going to be bad. The other limitation there is that landfills have to have sort of a structural integrity to them, and the waste material has to be compacted so that the structure holds its shape. And so if you put too much of an unstable waste material in a landfill in one location, it could affect the structural stability of that unit. And so the larger landfills are more likely to take infected carcasses. So one of the things we've been doing is to consider how we can sort of triage the carcasses, where if there are ones that we know are infected, we manage those on the farm. And if there are ones that we have tested negative, if we can send them to slaughter and they're safe and they'll pass the inspection and everything, well, that's the best thing you can do, because then it's not a waste material and you can use that valuable protein. If that's not possible and you still have, maybe you have a farm with 10 barns, one of the barns is infected, positive, and the other 9 are not, can you then send those non-infected ones to the landfill? And so that's what we're looking at, trying to work through those issues to minimize the amount of material that has to be managed as contaminated. And the other overall concern about waste management with ASF is that the farms have large buildings on them, and those buildings generally take up much of the acreage. And so there isn't a lot of space outside the buildings on that farm to do any kind of waste management on site. So if we're able to separate out that material that is contaminated and that which isn't, then it helps us to be able to dispose of the contaminated material on site and then transport the non-contaminated. Rendering is another off-site option, which is again designed specifically not just for generalized waste management, but specifically for carcass management. And it involves hauling the carcasses to this rendering plant where the material is dropped into a bin, if you will. It's then placed into a grinder, which pushes it into a cooker, and it drives off 60% of the biomass as a steam or water portion. 20% of the biomass is a fat or a tallow portion, which can be used as biodiesel or other products. And the remaining 20% is often used for pet foods or animal feed. If renderers, renderers are not anxious to take infected materials. Let me just say that, period, exclamation point. There may be rare instances where they could accept infected material or contaminated materials. And that's if their plant is unlucky enough to be within that 6.2 mile radius of the infected premises and they don't have any other business. But that's pretty rare. So that's not likely to be an option for us with the contaminated material, but it may be an option for the uncontaminated material. So we're working with them to figure that out. So on to the on-site options. Some of you may be familiar with the 2001 foot and mouth disease outbreak, where, traditionally at that time, and it's only been 20 years, but at that time, the traditional methods for disposing of carcasses were burning or burial. And it was in fact, it still is in the US regulations for foot and mouth disease. And so that's what the UK had on their books, and that's what they did. They started with burning. And the first photograph here on the left is one of the pyres that they had. And it takes a long time to take an animal that's 60 to 80% water and burn it down to ash. It takes a lot of wood and coal and anything you can throw at it to get it to burn and keep burning long enough to bring it down to ash. So these enormous pyres were built, and they were pumping out thick smoke for weeks at a time. And it was so bad they had to evacuate some communities, and it was not sustainable. So they had to stop doing that. And then they moved to taking the carcasses to a landfill. They didn't have room to put it in the landfill cells that were lined and already operating. So they created new pits right next to the landfill. They were unlined. They were just, there. They put all the contaminated carcasses in there. And to this day, they're still monitoring the contaminated groundwater that's coming from those mass burial sites. So, it's a real issue. The smoke from these pyres can be harmful to humans. There have been different technologies to try to improve the process, such as air curtain incinerators. This next picture, second to the left, is a trench air curtain unit, and the center right picture is a box air curtain unit. They operate similarly. The waste material is placed in the bottom. There's some fuel, like wood, put in with it, and then there's a natural gas that's ejected from this manifold system that fuels the flames, and then air flows over the top of it to keep the smoke down, kind of push it back into the trench or the box to keep the heat in, promote the burn, and minimize emissions. The problem is that you can't process a lot of material with these. So we used some of these boxes in 2015 high-path avian influenza response, and we were only able to process 5 tons per day per box. And previous slide, we were calculating 500 tons per farm in that North Carolina example. So, that would take quite a while. So, and speed is, critically important to contain the outbreak, but there are other limiting factors. So there are, there's also lots of other technologies, black boxes, if you will, gasifiers and different contained incinerators. Again, the throughput is the issue. And it may be difficult to get these permitted in many places. So in 2001, the UK estimated that every pig that was burned emitted 3 kilograms of particulate matter to the air. That's a lot. And the particulate matter causes human health effects like asthma, other respiratory disorders. And if the pyres are built with other material in place of wood, like tires and whatever else is laying around, the human health risks may be significantly greater. And then pouring too much liquid fuel on the pyre, like diesel fuel, also can result in soil contamination. And so what seems on the surface to be a quick, easy, cheap option could turn out to be an environmental remediation ongoing expense, which is exorbitantly expensive. There are some ways to mitigate the risks from open burning, though. And where you place the pyres at the distance from occupied areas, the size of the pyres can all help. And not burning trash or tires as well. In terms of burial, we're using burial to refer to a pit or a trench that's excavated 6 to 12 feet below the ground surface. The carcasses are placed in the bottom of the excavation, and then at least 3 feet of soil is placed on top to bring it back up to the ground surface. That's been used in the past. That's what people think of as the logical option. What they don't realize is that there's very little microbial activity 6 to 12 feet below the ground surface. And so we have encountered burial sites on farms that have been developed and other locations where when you dig into these pits 50 years later, you find the carcasses looking like they were just put there the day before. So there's very little decomposition that happens. They do lose a lot of the liquid, but the tissue doesn't break down. So the leachate is really a major concern with this option. So this is a cross-section of a burial site where you have the carcasses in the pit, and then the leachate is flowing from the bottom of the pit. It'll go down through the soil and eventually make its way to the water table. Eventually, that amount of time it takes and the concentration that it will be at when it reaches the water table depends on the distance as well as the soil types and the amount of moisture in the soil matrix. So there are regulations in the US about carcass disposal, and they require usually a minimum of 2 to 3 feet of distance between the bottom. I'm seeing some surprised looks. Yeah, 2 to 3 feet is the requirement from the bottom of the burial pit to the water table. And so, and then the other standard, the minimum standard we found in looking at the regulations for the 50 states, a minimum of 100 feet to the private drinking water well. So can you imagine if you're 100 feet from this pile and it's only 2 or 3 feet from the water table? And well, so they had that situation in South Korea in 2011. They had a massive FMD outbreak. They used burial. It was November, it was the rainy season, 4,500 burial pits. They had to excavate them. Immediately, there were about a third of them had to be dug up immediately, and they literally had leachate running from people's kitchen faucets. So, you know, it's one of those things. How sure are you that you have enough distance in this equation and the right soil types to prevent the leachate from getting to the water table? And it is important because of the high levels of nitrogen and phosphorus in the leachate, which are 1,000 times more than the standard for drinking water. And if there's an excess of nitrates in drinking water, it can be fatal to infants. And then there's the question of, do viruses survive in leachate? And so we only have one data point, and this was from 2015 HiPath AI in Wisconsin, where they had an infected poultry farm. The USGS and USDA went out and collected samples from the soil and groundwater, just generally on the farm, beneath the manure pits that they had, and they found, so it was PCR positive for many of the samples, meaning they found RNA from the virus. One of the samples also came up positive for virus isolation, and that was collected in groundwater within the first week of the farm being positive. So that suggests that the viruses can survive for some time. We don't know how long in groundwater. And if that groundwater is then pumped out and used to water susceptible species, you can perpetuate the outbreak. So, you know, the agricultural community is trying to figure out better ways to do this. And one of the things that surprisingly has been effective is composting. 85% of the birds that were infected in 2015 were composted. So it's 42 million birds. I would never have believed it if I hadn't experienced it. So the composting process involves laying a 1 to 2 foot thick layer of carbon, like wood chips, on the base, on the ground surface, placing the waste material, so the infected poultry or pigs or what have you, and then covering it over with another 1 to 2 foot thick cap of a maybe wood shavings, or it could be a finer material for the cap. And as the microorganisms in the waste material consume the tissue, they generate heat. That heat rises and it pulls oxygen in from the bottom, and so keeps that process aerobic. But it can't be too wet or it won't be aerobic, it'll go anaerobic. So there's quite an art to building these piles properly. And when they are built properly, they'll heat up to between 140 and 160 degrees Fahrenheit for a long period of time, which is plenty hot to kill many viruses. We have data from a number of sources. I'll show you after this slide. So there are a lot of upsides to composting, and there are some downsides. You know, it is a process that isn't disposal. You know, it produces a beneficial end product. So it's not completely wasting the protein. In Minnesota, where it was predominantly turkeys that were affected in 2015, the finished compost had such a high nitrogen concentration that people were clamoring for it to spread on their fields. Normally, finished compost is a good soil amendment. It improves the texture, but it usually doesn't have a lot of nutrients. And in this case, it did. And so it was really a valuable commodity after it had gone through the cycles of heating. And we required, APHIS, USDA required that the compost pile went through 2 cycles of heating. So it had to reach at least 120 or 110 for, okay, so we were using the the land application standards, 131 for 72 consecutive hours. And then, you know, there were some situations where we couldn't reach that. And so we were able to use like 110 degrees for 10 days, and we did daily monitoring of these piles. So it was extremely labor intensive, but it was extremely effective. And these are just some pictures of in-house composting for the poultry. This was one farm. It was an egg-laying farm that had 5 million birds, and they had enough property where they could compost on site. And the compost windrows could be seen from space and covered 6.5 miles of windrow length. So this is massive. And so when you think about the swine farms, you know, so 5 million. So we sometimes work in like cow equivalents. So one cow is 5 pigs and like 20,000 chickens, something like that. So, you know, that's 20,000 chickens, you know, so 5 million, if you did your pig equivalents, you know, that would be, we would be into the miles of windrows with composting pigs if they could, if the farm had enough space, which is unlikely. And the other downside, well, so some caveats with it are, it requires expertise to do this. We found that, you know, whenever a state would have its first new premises break, we would offer them, we'd say, okay, what kind of disposal, you know, what do you want to do? Well, we want to use composting. Okay, can we send you an expert? No, we've got this. Within 3 days, they're like, please send us an expert. And if they didn't use the experts to oversee the piles wouldn't heat up, and they would have to go back and rebuild the piles, which was, you know, just a waste of time. So we found that it was much better to send the folks with the expertise to oversee that. And the other thing is carbon source. If you're in the arid Southwest, you're not going to have necessarily enough carbon. Now, you know, we were able to use a lot of different materials, such as sunflower seed hulls in South Dakota and corn stover in Iowa. So, you know, there are other products that could be used, but there may not be enough for the amount of material that has to be processed. But there have been some studies, you know, so one of the first questions is, well, if burial is bad, doesn't it release leachate as well? So Iowa State did a study where they found that in the soils in Ames, Iowa, at least, the leachate, as measured by changes in the soil chemistry, only went about 4 feet below the ground surface. So that would suggest you needed a separation of 4 feet between the bottom of the compost pile and the water table. Cornell did a study on roadkill deer, and they composted them whole. So there's a difference with speed. When you have smaller animals like chickens, they'll compost in a month, whereas if you have a whole pig or a whole cow, it could take 6 to 12 months for that entire carcass to decompose. And even after a year, you may still have large leg bones with the cattle. And even with the pigs. So Cornell University did a study on deer, and they composted them, and they looked at a number of very hardy pathogens, and basically found that by 12 months, nearly everything was, you know, at least 4 to 6 log reduction, and after 18 months, everything was down, you know, to virtually non-detectable. So that was with the whole carcasses. Canada did a study with composting and FMD virus at their Winnipeg lab, and they found that within 10 days, the FMD was all inactivated in the pigs in the lab. We did a study recently in North Carolina where we were comparing grinding of pig carcasses with whole carcasses, and then subjecting them to composting or above ground burial, which I'll explain in a minute. And here are pictures after 8 weeks with the ground carcasses in the compost pile. You couldn't really identify what the tissue was. It was basically fully decomposed. Whereas with the whole carcasses after 8 weeks, we were still able to identify large pieces of tissue. So it significantly accelerates the composting process. And that is really important, because if we had ASF and if a pork producer chose to compost on site, their premises couldn't be released from quarantine until the composting process finished. And so if that could be 6 months to a year, that's unsustainable for a producer to survive that amount of downtime. And so the question was, well, if we could grind the material, would that speed up the process enough so that they could get back in business faster? And the answer is yes. But then the rest of the question is, can it be done safely? So does the grinding process produce aerosols that are going to spread the virus and make things worse? And so we've teamed up with some folks at the EPA Research Triangle Lab who did this awesome array of filter units and rigorous air monitoring. We are the worst reading we got, 250 feet downwind from the grinder. We had one hit that was 8 copies of pig DNA in the filter, okay? So now we're trying to figure out how to convert a copy of pig DNA into infectious dose. And so we've got modelers and our risk assessment folks at SIA working on that problem. Based on my back of the envelope calculations, I think we're a couple orders of magnitude below an infectious dose, but we'll let the smart people actually do the calculations. So what that means is, if we can grind safely and it speeds up the disposal process, it takes less space and can get the producers back in business, then that helps protect their livelihoods. And we'll get them back into production faster and contain the virus faster. So above ground burial is the last item I wanted to touch on. This is a hybrid between shallow burial and composting. So it involves digging a 2-foot deep trench, placing a foot of wood chips in the bottom of the trench, laying a single layer of carcasses on top of the wood chips, and then covering it with the soil that was excavated, planting it with grass, and leaving it there for a year, after which time it's pretty much gone. There's a little asterisk next to that. So this is an emerging technology. It's been tested in a number of locations, representing a number of different soil conditions and climatic conditions, with mostly good success. But we've had some issues with like severe rainfall events and flooding, which have caused it to be anaerobic and not break down. We have one study going on in Oklahoma right now, where we used whole sows. We inoculated their bone marrow with an ASF surrogate. We're using a swine pox. There aren't any really good surrogates, but swine pox was once classified in the same family with ASF, so we use that. After 2 weeks, there was no sign of viable pox virus. Now we're doing some lab comparisons to make sure that it wasn't just because the swine pox didn't like the bone marrow. We want to make sure that it's really effective. So that's happening in the lab. We're also testing soil quality below the piles. This was 100 sows, so it's a pretty large scale test. After the 2-month period, there was a spike in ammonia at about 30 inches below the plot, but by 4 months, that was back to background levels. So, and it was that spike of ammonia wasn't seen at deeper levels. So it appears it has a shallow minor effect. And then the third part of the study is looking at insect activity. The concern being that the insects might serve as fomites. And so we have their entomology department out there trapping and classifying bugs. We did have one beetle maggot come up positive with PCR, and we're trying to isolate the virus. And I don't know where that stands, if they were able to get enough material for that or not. But that's ongoing and will end up later this year. And that's a picture of one of those above ground burial pits in Oklahoma when they were doing the 2-month sample collection. They dug down into the carcass and they took a picture. And so this was basically the internal organs at the bottom here. This is the rib cage and the hide stretched across the top. And you could see that there had been, a significant amount of decomposition at 4 months. At the most recent testing, they found that the entire carcass had compressed at least 8 inches. So there's been significant decomposition there at the 4-month mark. And this is a photograph of the ground material placed in an above ground burial plot after 8 weeks. And there's a lot of white on that, and that's fungus. And so that's showing that there's active decomposition going on. And the folks who are most familiar with this technology say that it goes about twice as fast as normally. So if you would expect a whole carcass to take a year, then they would estimate it'll take about 6 months to fully decompose. This is just a chart to kind of compare the different options based on some of those, cost, time, and risk. I won't belabor that. And this was just another way of expressing summary of what seemed to be the options for ASF. That compost was likely to inactivate the virus, above ground burial likely to inactivate but still being validated, deep burial unlikely to inactivate, and so on. And I won't dwell on the storage and carcass handling and transport considerations, except to say that strict biosecurity is essential. And then I have a few more slides here of the grinders. There's different types. Well, we considered the vertical or tub grinders, but those are batch processes, which means you put in a cow, you put in a bucket full of wood chips, you grind it, and then you pour it out, versus a horizontal grinder, which is a continuous process where you can continue to load wood chips and carcasses continuously, and it will process and be discharged out the other end. And so this was the process that we were doing the air monitoring on. I already mentioned the 8 copies of pig DNA. And that's where we are with it. So I'm happy to take questions.
Thank you so much for your presentation. And as we like to thank all our speakers here at the One Health Academy, we have a gift for you, a flag that has been flown over the Capitol. So thank you so much for sharing your information with us this evening. I will now pass around the mic to anyone that has questions.
So I had one question for you. I know that when we had high-path avian influenza, part of the problem, even to getting to carcass management, was how do you euthanize that scale of animals in a timely fashion to prevent them from continuing to, replicate virus and potentially infect other farms in the area, and do it as humanely as possible. And I actually am not sure what the approach would even be for swine, and if that's something that you could speak to.
I can. So let me just caveat that by saying I'm familiar with the information. I'm not a veterinarian, but I can relay to you the information that I'm aware of. So it is a huge challenge. There are only, a certain number of options like captive bolt, electrocution. There's, drug injection, CO2 gas is another one. And so there are, limitations with many of them. So let's take the most sort of straightforward captive bolt. We've done studies that have determined you have to get the animal through a chute into a restraint, administer the captive bolt, check for that it's been effective, and then get the animal out of the restraint. And so it takes about 15 minutes per animal if you have like the most efficient setup you can imagine. And so when you do the math on that, I think, so we developed a calculator so people could go and put these scenarios in and do the math. And if I recall, when we put in 20,000 pigs, it was like 84 days. If you were running 2 sets of double lines with a total of 36 people operating this depopulation. Yeah, so captive bolt, not great. Electrocution is used in slaughter plants. It can be very effective, humane. In speaking with some colleagues in Lithuania, where they have ASF, they did have it in production, commercial production operations. The gentleman was telling me they had 164 pigs. They used electrocution. It took them half a day to put them down in their pens. It took them 2 days to get them out of the pens. And that was 164. So in, sort of trying to apply that to the US production systems, and, folks are talking about things like ventilation shutdown, which the AVMA has recently come out with their depopulation guidelines. Depopulation meaning that as much attention to animal welfare as possible under the emergency situation. So that doesn't mean it's perfect. They require the ventilation shutdown to be accompanied by CO2 gas, or, and it has to be accomplished within 3 hours. It's really not possible. Yeah, but that is what folks are saying. I mean, there are folks saying, we're just going to close the doors and walk away. So they're talking about tearing out walls of the barns, if they're using CO2 in the building, and trying to get the animals out. So, the hope is that those animals, that it can be caught early, that when we have stepped up surveillance, that, if we can keep an infected barn separated from any other barns on the premises and keep them from being infected, keep those animals ambulatory. If they do have to be depopulated, that they can walk out, and CO2 in trucks is an option that we've studied.
So something a little bit different. I'm curious to know if in any of the studies that involved like the soil or earth mounting or composting or burial, if you all or somebody at the Agricultural Research Service is checking on the soil health through microbiome, like what's happening to the soil microbes, because you see that spike of ammonia, which goes away, but are there long-term effects on the health of that soil afterwards?
So we were tracking E. coli was a sort of an indicator, so coliforms, which did increase, and we had baseline data. So once we have all the data compiled, I think that we could, look at that and answer that question. The studies that I'm familiar with, like the composting in Iowa, the soil's recovered, to baseline eventually.
Yeah, because you don't want to spread that E. coli around.
Thanks for the presentation. Quick question. Early on in the presentation, you showed a map of North Carolina and a lot of different pork farmers that in the area you could not bury. Why couldn't you bury in that map?
Thank you for asking that question. Yes, I meant to touch on that. So when NRCS does their modeling, it's based on some factors like how far is it to the water table, how sandy is the soil, Are there sensitive receptors? So is it near a stream, you know, a high class, you know, like a class 5 stream? Is it near drinking water? So those are factored into the rankings of the NRCS model. It also has to do with when they excavate the soil, if the soil will cave in or not. So if they can literally dig a hole or not.
Lori, thank you. Great presentation. I have a couple of questions. One, I want to make sure I'm interpreting the slides correctly. When you have the time, cost, and risk in pluses and minuses, does the plus mean that that factor is higher, or does that mean that it's positive and it's actually?
So,
For instance,
yeah.
cost. Does a plus mean it's high cost? That's a plus. That's a positive trait.
Yes. Let me get to one of them.
So I'll make sure I'm interpreting this.
Okay. So so a plus, in this case, for the above ground barrel, the plus minus means it's sort of middle of the road. So it's average time.
But if it was a plus time,
So it's a relative. So in terms of above ground barrel, the minus $ means a low cost,
oh, it does mean.
lower cost and moderate risk.
Okay.
Right. And it's summarized in that colorful table that I didn't spend much time on. And so again, in this one, for composting, it's moderate time, high cost, and low risk.
All right, thank you.
You're welcome.
You and also the other question I have is you talked about infectious dose. What is the infectious dose? How confident are we in that number? And are there any potential, in fact, you talked about concerns about getting into the environment. And are there any vectors that are known to be amplifiers after, as they could potentially carry?
So let me touch on the amplifier part. I am not a virologist, so I can't answer that one. There are vectors that will carry it, like feral swine could contract it and then carry it throughout if they had access to commercial herds. So that is a big concern. We do have a wildlife services group at APHIS who traps feral swine when they're hindering agriculture, and they test any of the animals that are captured. And I'm sorry, what was the first part of your question?
I just asked, what is the infectious dose and how confident are we with it?
So I've seen different articles. I'm using 100 plaque forming units, or is the TCID 50. But I've seen it as low as 10 or less, and I think it depends on the susceptibility of the host. And confidence is anybody's guess.
Any final questions? I'm going to close on the webinar, but do another question.
Thank you. Okay, thank you. My question is, you talked about just the massive swine population, using the North Carolina example, that would have to be processed. Would it be specifically a USDA operation, or would you be relying on private environmental consultants or other organizations to carry out the massive processing operation?
So at this time, we don't have any contractors that are trained to euthanize pigs. So any depopulation would have to be done in partnership between USDA and the producers and the state animal health agency. In terms of, so in terms of the disposal side of it, really, a waste is a waste is a waste. You have to characterize, what are its characteristics, and then you deal with it accordingly. So there are lots of environmental contractors out there who can do that type of work. And I believe our National Veterinary Stockpile does have blanket ordering agreements with 2 at this time. They periodically put out a request for proposals and, compete those awards.
Okay, everyone, thank you again for all her. I'll just start off with a few items. If anyone needs any more food on the way out, happy to take you go. And yeah, next month we will be meeting on second Thursday again. So look forward to seeing you guys. We'll send out emails with our speaker information. And yeah, thank you so much for joining us, and happy to see some new faces, and look forward to seeing you guys soon. Thank you and have a good night.